everybody. In this lecture, we are continuing our discussion of genetic regulation. In this lecture in particular, we are going to discuss how gene expression can be regulated at the DNA level prior to transcription even beginning. Epigenetics is when genes are regulated by chemical modifications that do not affect the genotype of a cell. This usually occurs via methylation or the addition of a methyl functional group to proteins involved in the storage of DNA called histones, or to individual nucleotides contained in the DNA. Let's look at both of these. DNA is a very precious molecule as it stores all of the instructions for life. Instructions for both the structures of an organism and their function. Those instructions are held in the nucleotide sequences of DNA, which are protected by having two strands bound by hydrogen bonds, which are then wound into double helix, which continues to wind and wind and wind and wind until it's effectively so tightly wound that nothing can access the nucleotides or instructions it stores. This is excellent for protection, but it's not really conducive to enzymes being able to reach the nucleotides in this DNA to read them into mRNA. Actually, when DNA is so tightly condensed, it effectively prevents enzymes from accessing the information stored inside, and thus this DNA is transcriptionally inactive. No genes in this tightly wound DNA are being transcribed because the nucleotides in this DNA are inaccessible. In order for DNA to be transcribed, DNA has to decondense. To understand how condensation and decondensation is regulated, it's important to understand how DNA is wound. The helical nature of DNA starts this process, but further coiling is facilitated by proteins called histones. Histones bind and wind DNA, and how tightly they do this has to do with the action of two complementary enzymes, methylases and acetylases. Methylases add methyl groups to histones, and this causes histones to bind and wind DNA very tightly. DNA that is heavily methylated and thus tightly wound around histones or condensed is called chromatin. Acetylases do the opposite. They add an acetyl group to histones, which causes them to have a looser association with the DNA, causing them to be less tightly wound. Since methylation of histones causes DNA to condense very tightly, heavily methylated DNA is not accessible to enzymes like RNA polymerase, and therefore it is transcriptionally inactive. In order for transcription to progress, RNA must bind to DNA, so the DNA would need to be decondensed. When DNA has less methyl groups associated with it, RNA polymerase can access the DNA to bind and allow transcription to progress. Such DNA is called active, referring to the fact that it is transcriptionally active, and is collectively called euchromatin. When DNA has more methyl groups associated with it and is thus tightly condensed, it's called silent or heterochromatin. Silent refers to the fact that this DNA is not being actively transcribed into mRNA. Methylation of histones is not the only mechanism by which transcription can be affected by methylation. In fact, transcription can also be affected by the methylation of individual nucleotides in the DNA. Methylation of individual nucleotides has a similar effect to methylation of histones in that it prevents transcription. Epigenetic modification of individual nucleotides typically involves methylation of the histones in a region of DNA. When this happens, RNA polymerase cannot recognize the cytosine, and thus transcription cannot progress. Thus, epigenetic modification of genes will result in genes being turned off. This is actually very interesting because it ends up leading to phenotypic change without a change in genotype. What this means is that the gene itself, the nucleotide sequence, remains the same. Like you can see here, in both instances, the sequence is ATTCCG. However, on the bottom, when a cytosine has been methylated, transcription does not progress, and therefore the gene is turned off. Interestingly, these epigenetic modifications can actually be inherited, meaning that if they are incurred in a mother or father, or even in a grandmother, they can be passed to offspring. Some of these epigenetic modifications are caused by microenvironmental effects, such as those that occur in a fetus and placenta during development. Some of these changes are induced by external environmental exposures. In fact, epigenetic modifications are believed to contribute to differences observed in identical twins. Some of these changes, such as those that occur throughout development, are homeostatic and contribute to our normal genetic variation. But some of these changes can be damaging and lead to disease. An example of homeostatic epigenetic modification is the methylation that leads to inactivation of one of the X chromosomes inherited by organisms with two X chromosomes. As you will learn in later lectures, the majority of cells in an organism contain two versions of each chromosome. Many aspects of phenotype, though, are based on a dose response of protein contributed from the combination of those chromosomes. In organisms with two X chromosomes, in order to maintain the appropriate dose of gene expression from these chromosomes, one of them is inactivated. This occurs via epigenetic modification. One of the X chromosomes incurs significant methylation and thus condenses into a tiny inactive state called a bar body. As expected, the structure is not transcriptionally active. Interestingly, in cats, fur color is one of the genes located on the X chromosome. Random X inactivation in these cats leads to the calico fur pattern. Some cells inactivate the X chromosome containing the gene for black fur, while some cells inactivate the gene containing orange fur. This is actually why all calico cats are genetically female. Only cats with two X chromosomes can have fur color impacted by random X inactivation. Oops. This is actually a really great video discussing epigenetic modification and X inactivation. I recommend checking it out, although we're not going to watch it right now in this lecture, just strictly due to time. While X inactivation is an example of homeostatic epigenetic modification, some epigenetic modifications are benign or downright harmful. Some environmental influences like tobacco or certain plastics, some insecticides, fungicides, herbicides, and sometimes exposure to mining and lead can lead to epigenetic changes that influence the development of certain etiologies of cancer. Environmental induced epigenetic modification can also lead to other health concerns. For example, exposure of a pregnant mother to tobacco smoke and other environmental toxins can lead to epigenetic changes during key developmental stages that lead to the fetus being predisposed to obesity and diabetes due to genetic programming in the adipocytes or the fat cells of the developing fetus. One of the most confusing things about epigenetics is how this can cause phenotypic change without genotypic change. So I wanted to clarify that a little bit more. To do this, let's revisit the processes of transcription and translation first under normal circumstances. You have a gene composed of DNA, that is shown here in blue. The 3' to 5' strand or coding strand of DNA is read into a 5' to 3' strand of mRNA via transcription, which is facilitated by the enzyme RNA polymerase. Since we have a T in the DNA, we have an A in the mRNA. Since we have an A in the DNA, we have a U in the mRNA, etc. Then this mRNA is sent to a ribosome where it is translated into a chain of amino acids linked by peptide bonds, aka a polypeptide or a protein. This is the central dogma of science. This is the process of transcription and translation and how we read DNA into the functional material of life. You learned already that genetic mutation is when something occurs that changes the genotype of a cell. For example, we can look at a missense mutation. Again, our normal gene is shown on the left, TAC, CCC, CGA, ATC, CTG. If a genetic mutation occurs, this changes the nucleotide sequence in the DNA. In other words, the genotype is changed. You can see on the right, we have a C mutated to a T. In some mutations, this has the effect of changing the phenotype as well. For example, shown here, the conversion of a C to a T led to a change in the codon GGG, which codes for glycine, to GAG, which codes for glutamine. Since structure equals function, changing the amino acid present in a protein can have impacts on that protein's function. However, the DNA code does have repetitive nature. Many amino acids have similar chemical properties. So some amino acid changes may not impact the overall protein structure or function. So in the case of a genetic mutation, you get a change in genotype, but you may or may not get a change in phenotype. It completely depends on the nature of the genetic mutation. However, with epigenetic change, you do not have a change in genotype. You can see here that compared to the normal gene, the sequence of nucleotides is the same. However, methylation of the cytosines in the gene leads to its inability to be transcribed into protein. This would have the obvious effect on phenotype in that there would be no protein. Thus, epigenetic modification changes the phenotype, but not the genotype. This slide just compares genetic mutation to epigenetic change. Again, genetic mutation causes a change in genotype. And depending on the impact of this change on protein structure, phenotype may or may not be impacted. However, in epigenetic change, genotype is unchanged, but methylation does lead to a phenotypic change. Both of these changes can be inherited if they occur in the germline or in cells that are passed to offspring. I also want to reiterate that epigenetic modification can occur at two levels, but with similar effects. Methylation of histones will cause condensation of DNA into transcriptionally inactive or silent chromatin. Methylation of individual nucleotides within DNA will lead to RNA polymerase being unable to read the gene into protein. So both lead to a gene being turned off. It is important to remember that epigenetic modifications are reversible though. So this is a form of genetic regulation. In other words, genes can be methylated to be turned off or demethylated to be turned back on. From this lecture, make sure that you know the terms epigenetics, epigenetic modification, phenotype, genotype, histone, condensation, decondensation, chromatin, methylation, acetylation, euchromatin, heterochromatin, and X inactivation. Make sure that you are able to compare and contrast the effect of epigenetic modification of histones and nucleotides. Be sure you understand the homeostatic function of epigenetic modification. Make sure you understand the impacts of environmental induced epigenetic modification. And be able to compare and contrast genetic mutation and epigenetic mutation. Thank you so much for watching this lecture, and I will see you next time.